Good afternoon. Welcome to the lecture series dealing with macromolecules. What we've done in the lectures to date is talk a bit about some basic biological chemistry, and the reason that we needed to do that was to eventually get to what I've been referring to as biologically relevant molecules. We talked about basic atomic structure. We talked about the structure of those atoms and how the electron configuration was important to allowing for chemical interactions to take place between atoms to build molecules. We discussed basic mechanisms or basic concepts behind chemical bonding. That is, we discussed covalent bonds, we also talked about ionic bonds, and we talked about hydrogen bonds. We then spent quite a bit of time talking about water. We also then discussed backbones for building biologically relevant molecules. And at the tail end of the last set of lectures, we also talked about functional groups and how those functional groups can be added to a basic carbon skeleton to build a biologically relevant molecule. So this lecture or lecture series is going to deal with macromolecules. Now, macromolecules are exactly what their name implies. The prefix macro comes from the Greek meaning large, and molecule, as we defined in the previous lecture, is two or more atoms held together by covalent bonds. So what we're dealing with macromolecules is extremely large molecules. These are very large in scale or in scope. When we talk about macromolecules, most macromolecules are what we refer to as polymers. And a polymer is a long molecule of many similar or identical units that are linked together by covalent bonds. In a polymer, you also have the root monomer, which is the single unit that is repeating over and over again in the polymer. So again, most macromolecules fit into this basic description as far as structure is concerned, being a polymer. Monomers are individual units that are linked together, one right after the other, to form the polymer. The best example or analogy I like to use for a polymer is a strand of pearls. A strand of pearls is a pearl after pearl after pearl after pearl. The individual pearl is a monomer, and the strand of pearls where they're all linked together would be representative of a polymer. Now, for polymers, the monomer does not need to be identical, but they do need to be similar in structure, and we will see some of those examples as we continue on with the lecture today. So to add to that analogy of the strand of pearls, there are, in some situations, different color pearls. Those could be different or distinct types of monomers that are, in fact, similar overall, but just differ in color. And that too could be a representation of a polymer. Now, another unifying feature of polymers is how they are synthesized and how they are broken apart. What you see in this particular slide is a generic example of the making and the breaking of polymers. On the left-hand side, you see a representation of synthesizing or making a polymer. Each one of the purple dots represents an individual monomer, and what you have on that left-hand panel is a short polymer that has 3 units, one right after the other, covalently bound to one another. And in this case, what we're going to do is add the next monomer onto the chain to synthesize a little bit longer polymer. And this is how polymers are in fact synthesized, typically one monomer at a time. Now, there are some exceptions to where it can be more than one monomer at a time, but in most cases, it's one monomer at a time. The name of the reactions that synthesize polymers are known as dehydration reactions, also known as condensation reactions. Both of those terms that describe the reaction for the synthesis of the polymer involves the removal of water. So again, if you look at the left-hand figure, you can see the hydrogen from the short monomer and the hydroxyl group from the monomer being added on. Let me say that again. The hydrogen highlighted in the short polymer and the hydroxyl group in the unlinked monomer are going to come together to form water. Water is released in these reactions, hence the term dehydration reaction or condensation reaction. Dehydration reaction or condensation reaction. On the right-hand side is the representation of how polymers are broken apart. These are broken apart by reactions known as hydrolysis reactions. The term hydro refers to water, and the word lysis means to break apart. So hydrolysis reactions use water to break apart the molecule in question. So on the right-hand side, you see a polymer with 4 units, one right after the other. Water is going to be required to break apart that covalent bond where the arrow is pointing between units number 3 and number 4. And that is exactly where the covalent bond is broken, where the 1 unit is now removed, the 1 monomer is removed, and you still have a 3 monomer polymer left behind. And typically hydrolysis reactions occur in this way, 1 monomer at a time. There are, however, again, some exceptions where more than 1 unit can be broken off, but typically this is how they happen. And so again, these are unifying characteristics of polymers. There are 4 distinct categories of macromolecules. The first one we're going to focus on are proteins. Now, proteins are extremely diverse in their structure and function. They are, in fact, the most abundant macromolecule category in a cell. Proteins are polymers composed of 20 different amino acids, and we will look at the different amino acids in just a minute. What I want to highlight on this particular slide is, again, that they are the most elaborate and diverse of life's molecules. They actually fall into many different types of functional categories. What unifies proteins is the way that they are specifically synthesized and the fact that they are made up of amino acid monomers. And again, we'll highlight that in a few slides in just a minute. But again, what you can see from this slide are some of the categories or major types of proteins that exist. They can be things such as structural proteins that might add support overall to a cell or an organism. In this case, you're looking at the horns or hair in, again, that first example. They can act as storage molecules where they can store certain components inside of the cell or in the organism overall. They can act as contractile components that allow to help for movement in muscle overall. They can act in transport at the cellular level to help move molecules into and out of the cell. And probably by far the largest category of proteins are categorized as enzymes. Enzymes are molecules that help accelerate chemical reactions, and we will spend quite a bit of time talking about enzymes a few lectures down the road. Proteins, again, are polymers, and they are polymers composed of 20 different amino acid monomers. What you see in this figure are 2 amino acid monomers. What I want to do is just focus on one of them so that you get an idea of the structure of a typical amino acid. A typical amino acid has what's highlighted in the green, which is the amino group, one of the functional groups that we talked about in our last lecture. There is also highlighted in the pink the carboxyl group, which is also another component that we discussed, or structural functional group that we discussed at the end of the last lecture. And then you have at the center of this molecule something that is referred to as the alpha carbon, and it is the alpha carbon that the amino group is covalently linked to, as well as the carboxyl group is covalently linked to. In addition, underneath that carbon, highlighted in the yellow, you see the word side group. This is also referred to as the R group or the side chain, and in the next slide we will highlight those in a little more detail. So this is the basic structure of an amino acid. There are in fact 20 different amino acids that can be found, and these are what act as monomers to synthesize the protein polymer. What you see again in this figure is an example of the dehydration reaction or condensation reaction that will lead to linking two amino acids together. In this case, you have the hydroxyl group from the carboxyl group in the pink on the left-hand side, and you have the hydrogen that's part of the amino group from the amino acid on the right-hand side, and those components are going to be what come together to form the water in the dehydration reaction. There is a covalent bond that now links those two amino acids together, and the specific name for this covalent bond is referred to as a peptide bond. I want to add a few additional definitions at this point. The true term for a polymer of amino acids is a polypeptide. When we use the term protein, a protein is one or more polypeptide that is folded into a specific functional conformation, and we're going to talk about how that happens in the next series of slides. But again, it's important to distinguish the term polypeptide from the term protein. Polypeptide is the polymer of amino acids. Protein has function associated with it. What you see in this slide is a figure that represents all of the 20 distinct amino acids that can be utilized to synthesize a polypeptide, which can eventually result in a protein. I won't ever ask you all 20 of the amino acids by structure or by name. If you take more advanced classes and perhaps biochemistry, that might be the point where you'd have to memorize all of these. But for our purposes, I don't need to worry, or I don't need you to worry about the 20 of individual amino acids. What you do need to know and be aware of are what's highlighted in the different colors. So in the different colors, what's highlighted are the R groups or the side chains that distinguish or differentiate each amino acid from the other. What you should also notice when you look at this particular table is that they are grouped into categories. Those side chains that are highlighted in the green are nonpolar side chains. You have to go back to our biological chemistry lecture to understand what nonpolar means. What that means is that they are dominated by nonpolar covalent bonds. What you should then think about is, what does that mean for the interaction with water? We discussed the term hydrophobic and hydrophilic a few lectures back, and we discussed the fact that things or molecules that are dominated by nonpolar covalent bonds are going to not interact with water readily. Those things are going to typically, or those molecules are typically going to be hydrophobic. The next group down, highlighted in the pink color, are polar side chains. These are side chains, or R groups, that are polar in nature, meaning they have typically polar covalent bonds. In this case, they behave the opposite of the nonpolar side chains, because polar side chains, molecules that are polar in nature, are going to interact readily with water and be hydrophilic. The last two categories, indicated by the orangish color and the blue color, indicate acidic side chains and basic side chains, respectively. These are side chains that have net positive charge or net negative charge. The net negative charge are going to be the acids, and the net positive charge are going to be the bases. These two are going to be side chains that will in fact readily interact with water because of the positive and negative charges that are present. So those are the things that you do need to know, is the different categories of amino acids and how those categories dictate their behavior with water. So I highlighted for you on a previous slide the differentiation between the term polypeptide and protein. When we talk about a protein, it is a polypeptide that has been folded into some final functional conformation. A protein has function associated with it. What we need to do then is discuss protein structure. How is it that we end up with a protein, a functional molecule, overall? So there are 4 structures associated with protein, or 4 levels of structure associated with proteins. The primary structure is the specific sequence of amino acids overall. It is the arrangement of these amino acids that differentiates protein from protein from protein. So in this figure, really simply, you have at position number 1, the first amino acid that would be synthesized or utilized in synthesis. And you can work your way down the sequence all the way to the bottom where amino acid 129 has been identified. So this is the primary structure for this particular protein. It is the sequence of amino acids that are linked one right after the other. The next level associated with protein structure is referred to as the secondary level of structure. The secondary level of structure forms specifically because of hydrogen bonding between amino and carboxyl groups in the amino acids that make up the protein in question. The amino and the carboxyl group are considered part of the backbone in structure, and specifically when hydrogen bonds can form between peptide chains, this is what allows for the formation of the secondary structure. You can see that hydrogen bonding in the left-hand figure. And on the right-hand side are the 2 categories that can form, or 2 structural categories that can form because of this hydrogen bonding. So the secondary structure is due to the hydrogen bonding, but results in either the formation known as the alpha helix, which is the helical shape that you see, or something referred to as a beta pleated sheet. The helix is something that would look like a spiral staircase, if you can envision that in your mind. A beta pleated sheet is if you took a paper and folded it back and forth, sort of in an accordion fashion. This is what a beta pleated sheet would look like. So hopefully you get a feel for that structural features that distinguish the alpha helix or the beta pleated sheet. Regardless of what the formation of those structures are, they are due to hydrogen bonding between the amino and the carboxyl groups in the backbone of the structure. After the secondary level of structure comes the tertiary level. In the tertiary level, what you have is more complex 3-dimensional structure formation due to lots of different types of interactions. But specifically, these interactions are between the side chains of the amino acid groups that make up the structure overall. What you can see highlighted in this slide are lots of different examples of these interactions that form or can form between the R groups or the side chains. The first one in the top left would be hydrogen bonding that can occur between 2 distinct amino acids. In the center, you can have what's referred to as van der Waals interactions, which really just is a hydrophobic interaction where this portion of the chain of amino acids is going to tuck away from water. Next, you have what's known as a disulfide bond or a disulfide bridge. When we talked about functional groups, again, at the end of the last lecture, one of the functional groups we discussed was the sulfhydryl group. And I said that it's going to become important when we talk about protein structure. The disulfide bond is in fact a covalent bond that forms between sulfhydryl groups of different amino acids. So in the figure you see that covalent bond linking the sulfhydryl group between 2 different amino acids in different positions of this stretch. Lastly, you have ionic interactions that can form. And ionic interactions, like the ionic bond that we discussed in our previous lecture, is going to be the attraction of positive and negatively charged ions. These are going to be those components that we talked about that are acidic and basic overall in the table when we discussed amino acid structure. So what you can see here is there are a variety of different ways that tertiary structure can form, and it is the specific placement of amino acids that allows for the three-dimensional shape to eventually form due to all of these interactions that you see here. Lastly, we can talk about the quaternary structure. Remember, I defined the term protein as a polypeptide chain that has been folded into its final functional conformation. That can be as simple as those levels that we just discussed, but there are some proteins that are functional only when multiple polypeptides come together, or more than one polypeptide comes together to give the final functional protein. And in that case, that's what you have with the quaternary structure. It is when you have more than one polypeptide chain coming together as a complex to form the final functional protein. There are two examples that you see in this particular slide. The first one is collagen, where you can see three distinct polypeptides come together to form the collagen molecule. We also have on the right-hand side hemoglobin, and hemoglobin has four distinct polypeptides coming together to form the final functional hemoglobin molecule. So again, that's the example of quaternary structure. Not every single protein requires a quaternary structure, but some in fact do, and so we have to discuss it here. So how does all of this happen? How do these proteins fold into their final functional conformation? Well, it turns out that the number one answer is actually spontaneously. It is the position of the amino acids in the primary sequence level that then dictates the secondary structure, and that dictates the tertiary structure, and can also play a role in quaternary structure where that is necessary. So how does a polypeptide fold? Again, most fold spontaneously due to the position of specific amino acids. There are, however, an additional set of proteins found inside the cell known as chaperonins. And a chaperonin is a complex that looks kind of like a hollow barrel with a cap. What you see in this figure down at the bottom is that the unfolded polypeptide is threaded into the barrel. The cap is then put on, and it is this placement inside a confined structure that then helps with allowing for the final tertiary structure, tertiary or quaternary structure of the protein to come about. So again, some fold spontaneously, some get a little help, and that's what you see in this particular slide. The next macromolecule category I want to discuss are nucleic acids. Nucleic acids include both DNA and RNA. These are information storage or information containing molecules. They provide the directions for building proteins, but they also provide directions for everyday function inside of the cell. The typical flow of information is like what you see here on this slide, and towards the tail end of our semester, we will look at this mechanism in much more detail. But for now, suffice it to say that DNA is the genetic storage molecule. It contains all of the information or instructions for a cell to survive. Typically, what happens with that flow of information is genes are copied on a gene-by-gene basis in a process known as transcription to produce an RNA molecule. So an RNA molecule is a copy of a gene found in the genome of an organism. In most cases, the RNA is then used as instructions to synthesize a protein that the cell needs. So again, DNA and RNA are information storage molecules that contain information and instructions to synthesize proteins that the cell needs. There are some other examples where Proteins are not directly synthesized, but for our purposes, we'll leave it at that. So nucleic acids are made up of nucleotide monomers. So the monomer, the individual unit in a nucleic acid, is a nucleotide. As I mentioned on the previous slide, the two categories of nucleic acids are DNA, which is short for deoxyribonucleic acid, and RNA, which is short for ribonucleic acid. What you see on this slide are examples of monomers of nucleotides utilized in the synthesis of either DNA or RNA. In general, the structure is very similar, but I'm going to highlight some differences in just a minute. The similarities between the nucleotides for DNA and RNA are that there is a phosphate group, a 5-carbon sugar, and a nitrogen-containing base in every single nucleotide. What I want to do is we'll highlight some of those differences on the next slide. Okay, so in panel A, you have a generic nucleotide structure. Every single nucleotide, regardless of whether it's going to be utilized to synthesize DNA or RNA, has a phosphate group, which again is one of those functional groups we talked about at the tail end of the last lecture, and that's highlighted in the yellow in panel A. You also have in every single nucleotide a 5-carbon containing sugar, which is highlighted in the center of panel A in orange. And lastly, in green, you have a nitrogenous base, which just means it's a molecule that contains nitrogen in it and typically acts as a base. So in panel B, what I want to do is highlight the differences in the sugars that you can find in DNA or RNA nucleotides. So there is a difference in what type of sugar is found in a DNA nucleotide versus an RNA nucleotide. On the left-hand side in panel B, you have ribose, which is the sugar that is found in RNA nucleotides. And on the right-hand side of panel B is deoxyribose, which is the sugar that you find in DNA nucleotides. The only difference is highlighted in the little pink square. Ribose has a hydroxyl group covalently bound to carbon number 2, and deoxyribose is missing the oxygen, hence the deoxy in the name, in that carbon number 2 position. So that's the only difference between the two sugars, but they do differentiate DNA nucleotides from RNA nucleotides. Also, on the right-hand side, in panel C, you can see the different categories of bases. There are 2 broad categories of bases. There are pyrimidines in the top row, which are 6-membered ring structures, and purines, which have a 6-membered ring fused to a 5-membered ring. You do need to know the difference in basic structure between a pyrimidine and a purine, and you do need to know which specific bases fall into which category. Pyrimidines are cytosine, uracil, or thymine, and purines are guanine and adenine. These bases have a single letter abbreviation that is oftentimes utilized when talking about sequence in DNA or RNA. The other difference between a DNA nucleotide versus an RNA nucleotide is the presence or absence of one of these bases. Cytosine and thymine are found in DNA-containing bases, whereas cytosine and uracil are found in RNA-containing nucleotides. So uracil and thymine are replaced for one another. Uracil is found in RNA nucleotides, thymine is found in DNA nucleotides. Otherwise, the others are present in each of the categories. So as I mentioned, this category of macromolecules is known as nucleic acids, and these are polymers that are made up of nucleotide monomers. The synthesis of a nucleic acid occurs in that same generic way that we talked about a few slides back, with condensation reactions or dehydration reactions. In the left-hand panel, what you're seeing is the condensation reaction or dehydration reaction linking two nucleotides together. The bond that forms between two nucleotides is specifically referred to as a phosphodiester bond. Just like the bond that linked two amino acids together had a special name, a peptide bond, the bond that links two nucleotides together has a special name known as a phosphodiester bond. And in the panel on the right, you can see linkage of several nucleotides, one right after the other, giving you what is known as a polynucleotide. A polynucleotide, like I showed you on the last slide, was a linkage of nucleotide after nucleotide after nucleotide. There are going to be some subtle differences then in structure overall of DNA versus RNA. RNA, not shown on this slide, but RNA is typically always found as a single-stranded molecule. It is a polynucleotide, meaning there are nucleotides linked one right after the other in the RNA structure. It is, again, typically a single strand. Now, it can fold into complex shapes and structures, but overall it is still a single strand of nucleotide after nucleotide after nucleotide. DNA can form, or needs to begin with, the polynucleotide structure, and that's what you see on this slide in panel A, is a single strand of DNA. This is the nucleotides linked one right after the other. This is a very simplified view of a polynucleotide DNA strand, just to highlight the sugar phosphate backbone and where the bases are. Again, if you go back and look at the nucleotide structure, you can identify the sugar and the base and where the phosphate group is also located. So again, the sugar phosphate portion is referred to as the backbone, and the nucleotides sort of stick out. In the complex functional form of DNA, DNA is a double helix, meaning two single strands of DNA come together in a complex association and forms the helical shape. So hopefully you can distinguish those two strands from one another in panel B. Lastly, I want to highlight DNA structure in a little more detail. If you look at the left-hand panel, what you see are two single strands of DNA that are interacting with one another where the bases are. So in the red in this figure, you have your sugar phosphate backbone, like we highlighted in the last slide, and you have those bases pointing in towards one another. So if you could split this figure totally in half down the middle, you would have one polynucleotide chain and another polynucleotide chain. The way that they do in fact interact is hydrogen bonds form between the bases of neighboring nucleotides to allow for this complex interaction. So if you look at the figure on the right, the first one, where it shows the cartoon of base pairing, what you have is a structure that looks kind of like a ladder. Again, you have the sugar phosphate backbone of each of the single polynucleotides highlighted in the red. And then you have the bases interacting with those hydrogen bonds, making what looks sort of like rungs of a ladder, the parts you would step on a ladder. If you now took that structure and twisted it, that would give you the classic helical shape in the double helix. It's a double helix because two single strands of DNA come together and interact and then are twisted in the helical shape. So this is where I'm going to stop today's lecture. And the next lecture, part 2, we will continue on with 2 additional categories of macromolecules.